Hello, everyone. My name is Marian Galovic. It's a pleasure to be here to talk to you about VBM and SBM. These are two acronyms standing for Voxel-Based Morphometry or Surface-Based Morphometry. I am the head of the Epilepsy unit at the University Hospital in Zurich. Let's dive straight into it. So let me first talk about an interesting study that's been published some time ago. So maybe some of you have been to London, and maybe some of you took a cab in London. So these are not taxis, these are called cabs, and they do not use a sat-nav. Why is that? Because cab drivers are required to know the navigational aspects of London by heart. This is called the knowledge, and these cab drivers study for a very long time in order to gain this knowledge. So there are 9 million people in London, more than 600 landmarks, 25,000 streets, and it takes 2 to 4 years to really gain this knowledge. So it's like placing an atlas of London in your brain. And now what did researchers find? They found that there's a navigation-related structural change in the hippocampi of cab or taxi drivers in London. So what did they do? They scanned these taxi drivers or cab drivers while they were acquiring the knowledge and compare them to healthy individuals or normal people who weren't cab drivers. And they found that licensed London cab drivers had an increased gray matter in the posterior hippocampus, more on the right than on the left here. And this was related to possibly areas that are important for spatial memory and neuronavigation. And they received the Ig Nobel Prize for this study. It is not such a stupid study as it sounds. So they also used later medical doctors as subjects, and they found that these people did not have these increases because medical doctors, they learn and study a lot, but they do not need these navigational aspects compared to the London cab drivers. Also bus drivers, they drive the same routes over and over all the time. So they don't have these navigational changes in their hippocampi. So therefore, it seems like this is really neuronal plasticity happening in the brain that can be analyzed using advanced neuroimaging. So which method did they use to do this? This is a method called Voxel-Based Morphometry. So what does this mean? What are voxels? A pixel is a two-dimensional thing, whereas a voxel is a three-dimensional thing. It's a volume. Usually it may be a cube, but it may also have a different shape. So voxel-based morphometry is measuring the morphometry of the brain, sort of how the brain looks like, based on voxels, so based on little volume cubes. And this is what actually the data behind an MRI scan usually looks like. So it's coded like a three-dimensional matrix, and each of these little cubes, each voxel, has a certain intensity attached to it, which tells you what intensity the image program should display. Now, how do we compare different brains together? Now, this is something that the investigators of the hippocampus of the Ig Nobel Prize study have done. And how do you compare different brains together? They have different shapes, they are in different positions, and each brain is slightly different. And you want to bring them into a certain sort of standardized position, standardized template, we call it, in order to make each place in the standardized template correspond to each place, to the same place in each of these brains that are comparing together. And that is a major problem. So what the software actually does in order to get this is that it shifts around bits and pieces of the brain and tries to realign all of them so that all voxels are in the same place compared to the template. And then you can compare voxels, all regions together, because they correspond to the same areas of each person's brain. So how is this being done practically? Now, when you have here the original image, first, usually the programs that are being used for voxel-based morphometry do a process that is called segmentation. That means that they split the signal into the signal that corresponds to gray matter for white matter, CSF, and also other tissues in the brain. And the next step is classically called normalization. So then you try to wiggle around these parts of the brain and to place them all in the same position so they correspond to the same areas on the template brain. Right. Usually use a specific atlas. We typically, in voxel-based morphometry, the MNI atlas, which has been developed and the Montreal Neurological Institute is being used. And according to this atlas or to this template, then each sort of cube in a person's brain after normalization corresponds to the same cube in another person's brain, to the same region or area of that person's brain. And then you can do statistics. So what happens with statistics, for every voxel, so for every little cube, you get the gray matter volume inside this voxel. And for every voxel, you have actually a number for each subject. Then you just compare these numbers between groups of your subjects. For example, one group is the cab drivers and the other group is healthy volunteers comparing to the cab drivers. And you run a statistical test for each of these voxels. Usually the general linear model is being used for these types of statistical tests, but actually any type of statistical test could be applied for statistics. Now, a major issue is, as you can see, that the brain consists of many, many voxels. Usually it's around 500,000 voxels. So it's a huge number of statistics that you're going to do. And this takes us to a multiple comparisons problem. So when you perform a statistical test for each and every voxel in the brain, you will repeat the statistical test 500,000 times. And with a p-value below 0.05, you can be certain that just by chance, 5% of these tests will come back as statistically significant. So in other words, you will have 25,000 false positive voxels in your brain. They want to prevent this. So there are different methods to correct for multiple comparisons. Later we'll see how these can be implemented in the software. So you need to correct for multiple comparisons in order to reduce the number of false positives results in your statistics. What else can you do? Another possibility is to reduce the number of comparisons. So you split the brain into larger chunks, into, for example, predefined regions of interest. And then you calculate the mean value of, for example, gray matter volume for each of these regions. And you can compare the regions between your subjects and between your groups. So this may reduce the number of comparisons from 500,000 to around 50 to 100, depending on the number of regions that you are being used. The problem is you also reduce the resolution because the regions are typically bigger. So you will not be able to pick up effects that affect only a small portion of a region or that are affecting several regions at once. And this sort of may impact how your results will look like. When you run your statistic, this is now on voxel-based data, so on these little small cubes of data, you will get a statistical parametric map. That is actually a map of your statistical results. And then you can threshold it to only see the statistically significant results. And that means that you sort of mask all results that do not meet your statistical threshold. You also, in this process, try to adapt for multiple comparisons. So you do not include false positive in your results. And this creates a map of statistically significant differences between the groups that you've been comparing. Now, which problems are there? So a main limitation is that voxel-based morphometry splits the brain into little cubes. So you may be measuring the amount of gray matter in this cube, but you also cannot say if other problems may be causing differences in gray matter volume in your voxels. So this may be also due to differences in folding, in misregistration, so that the normalization process didn't work quite well, differences in contrast, or in misclassifications. For example, by including dura actually as part of the gray matter in a voxel. Another method that can be used to measure brain structure is surface-based morphometry. So what's the difference? In surface-based morphometry, you're trying to extract the surface of the brain, and you're trying to extract the surface of gray matter, the PL surface of gray matter, and also the surface of gray matter that's the boundary to white matter. And in this regard, you get a sort of a column of gray matter in 3D, and you can measure the thickness of that column. So you're usually measuring things like cortical thickness using surface-based morphometry, but you could also measure other things like surface area. And I recommend you to have a look at a talk of Carolina Ferreira-Tuesta about FreeSurfer, where she describes this process in a bit more detail. So using surface-based morphometry, you are able to reduce some of the mistakes or potential issues with voxel-based morphometry. But you again have the issue that the brain is not a perfect circle, but it actually has the gyri and sulci, and that each brain has a different form and structure, and also size, and also orientation. So it looks like this deflated balloon. And what you're trying to do then with surface-based morphometry is to inflate this balloon, inflate sort of the brain surface, and then you can measure each point on this balloon that should correspond to the same point or same area in each person's brain that you're studying. This is the process that's being used. So first you extract the surface mask, and then you can inflate it to a sphere and normalize it based on the sphere, where every point on the sphere corresponds to the actual point in a subject's brain. What else is important? Smoothing also matters. So as you will see later, we do smooth this data. Why is that? If you have perfect data without noise, you actually do not need to smooth at all. But if you have data that's usually a bit noisy, and that is the case with neuroimaging data, then to improve your signal-to-noise ratio, smoothing may help because it helps with noise in your image. And this is shown here, where after smoothing, you get a relatively homogeneous image that reduces the noise in your image. Another issue is registration. Registration never is perfect, and that's why using smoothing, you try to help with these little misregistrations that happen when you analyze or when you pre-process your data. And the last thing is statistics, because most of the statistics being used are based on a general linear model. Then you want to have a Gaussian distribution of your data, and in this regard, smoothing also helps. Right, so this was a very short theoretical introduction, and let's do something more practical. Right. Let's imagine you want to calculate the differences in brain structure between patients with left mesial temporal lobe epilepsy with hippocampal sclerosis compared to people who are healthy, who do not have epilepsy. So this is a very standard problem, and it has been done in literature many, many times. But this is one of the most basic comparisons that I can show you. So I chose mesial temporal lobe epilepsy with hippocampal sclerosis, which is a very, very common disease in adults, and we have many scans like this in most centers. And I chose only those with left temporal lobe epilepsy in order to make this even more simple. Right, so what are previous results showing us? This is a large multi-center study, probably one of the largest multi-center studies performed in epilepsy, called the Enigma study. And this is a structural comparison of left MTLE data with hippocampal sclerosis versus healthy volunteers. And it's a bit of a confusing slide, so I'll walk you through this. So here are subcortical findings, and these are cortical findings. And this is left temporal lobe epilepsy. So you see that the left hippocampus here in red, it has atrophy. This is also the left thalamus, as far as I'm aware. And and also the basal ganglia have some atrophy on the ipsilateral side. Whereas on the contralateral side, there's no atrophy of the hippocampus. Looking at the cortex, there is this very widespread pattern of atrophy. And this already shows you that atrophy in temporal lobe epilepsy is not restricted to a certain part of the temporal lobe, but really affects large areas of the brain. It usually affects these areas in the pericentral cortex, precentral cortex usually, also in the frontal lobe, and also some areas in the temporal lobes, usually quite bilaterally, and also some areas in the precuneus. Right, so this is based on regions. What about if you look at it at surface-based analysis that really looks at each column of the surface? This is an analysis done at McGill University, at the Montreal Neurological Institute, and it shows you again very similar areas in both hemispheres, affecting the frontal lobes, the temporal lobes, and also some parts of the precuneus here. Right, so let us replicate these findings and do some sort of a similar analysis. So I pulled data of 78 subjects with mesial temporal lobe epilepsy, and this is the median age of these subjects. This is so that there were a few more females in this group than males. All of them were lateralized to the left. All of them had temporal epilepsy, and all of them had hippocampal sclerosis, as seen by the radiologist. And duration of epilepsy was very long. So the mean duration was 26 years. So it's a very long-term study, and all of them had some drugs, median 2 drugs. Some of them were operated at a later time point, and around half of them became completely seizure-free in long-term follow-up. Now compare this to 48 healthy volunteers, and the distribution of sex and age is largely similar with the mesotemporal lobe epilepsy cases. So let's first look at the hippocampus. So we know that all of them had radiologically verified hippocampal sclerosis. What can we do? We can also measure hippocampal volumes. We can do this using an automated method. And this is a study published in 2013 for epilepsia that described such a method, which is called HippoSec. And I'm mentioning this because it's freely available. So you can go to this nifty website which is a UCL product, and it can be used freely. You just put in your email address, you upload your T1 high resolution images of your subjects, and off you go. And it gives you back these nice reports of the volumes of your left and right hippocampi of the subjects that you've submitted. And you can also correct them for total intracranial volume. So what is the total intracranial volume? That is the volume of the intracranial mass. And you can imagine if you have a person that is relatively large, big, then they will have a big intracranial volume. And also the hippocampi will be bigger compared to a person that is relatively small. So that is why you need to correct for total intracranial volume in order to get sort of a relative size of the hippocampus compared to the overall size of the brain. Right, so we've done now hippocampal volumetry using HippoSec, and I'm showing you the results here. And so these are healthy volunteers, and I've put in the 95% confidence intervals. So obviously 95% confidence intervals, there will be a few outliers. So the 5% are the outliers. And here are people with mesotemporal epilepsy. And you see that the large majority of them has a hippocampal volume on the ipsilateral side that is lower than these 95% confidence intervals. That is 91% had a smaller volume. And some of them have relatively normal looking volumes, but obviously there are also other characteristics of hippocampal sclerosis besides volume that may lead to the radiological diagnosis of hippocampal atrophy or mesotemporal sclerosis. What else can you do? You can look at the 3D shape of the hippocampus, for example, using a software called SALT, which you can download on this website. And it's a bit tricky to use, but essentially what it does is it creates a 3D mesh out of your hippocampi, and then it wiggles them around, trying to trying to adjust them to look like a template hippocampus. And then you can again compare the surface of your subject hippocampus to, for example, a surface of a normal or a template hippocampus. And we've done this here in this regard, and we were looking at displacement in millimeters. So whenever this shows red or yellow things, that means that there is inward displacement. That means that there is some sort of atrophy, that the hippocampus is smaller or the surface of the hippocampus is inwardly displaced in these points compared to a normal template hippocampus. And that may point to atrophy. So looking again at the group of healthy volunteers here, you see mostly gray results. So there's actually no change compared to the normal template. And this is the group of left MTLE. And they have this displacement of between -1 to -2 millimeters displacement compared to the normal values. Then you can run a statistical test on this. So comparing the two groups, comparing the group of healthy volunteers with the mesiotemporal lobe epilepsy patients. And this displays the T values on the surface of your left hippocampus. And when you run a statistical test, you see that almost all of the hippocampus shows statistically significant atrophy. Right. And let's look at something what you can do yourself. Right. So let's look at a practical example of voxel-based morphometry and surface-based morphometry using SPM. So I hope that all of you have SPM running on MATLAB or in a similar environment on your computer. You start it easily by typing SPM. I'm sorry for my computer, which is a bit slower than usual. So now this opens up this window. Because we want to do voxel-based morphometry, we click on this thing. We wait for SPM to load. And I will be showing you mainly voxel-based morphometry and surface-based morphometry using a software that is called CAT12. So it's a computational anatomy toolbox, and that's a toolbox that you can use for SPM. Wow, this is really taking a long time, probably because I'm doing the Zoom recording at the same time. And you can download CAT12 as a toolbox for SPM. Right, let's wait for SPM to load. Sorry for this again. But this is what it usually looks like. Let me show you the website of CAT12. This is the manual of CAT12. CAT12 has an excellent manual, so that's why I really recommend you to use this software. It's relatively straightforward to use. It has an excellent manual which describes all of these steps in a lot of detail. And this is the main website of CAT12. This is what it looks like. And when you scroll down, you can download it either as a standalone version or as a toolbox for SPM. Right. And then it's very easy. After you've downloaded it, you go to the folder where SPM12 is on your computer. You locate the toolbox folder and you just drag and drop CAT12 into the toolbox folder. Then you can start it up. So I recommend you to use CAT12 because it's a software, a toolbox running on SPM that has been specifically developed to support voxel-based morphometry and surface-based morphometry, and the methods are really top-notch. It has a great manual, so it's very easy to use and integrates a lot of steps that are not in the standard version of SPM. You can also use a different software for surface-based morphometry. For example, you could use FreeSurfer, and there's a talk about FreeSurfer by Carolina Ferreira-Atuesta, and I recommend you to have a look at that. That's really the gold standard for surface-based morphometry. But CAT12 is, I would say, at least as capable as FreeSurfer in many regards. It requires less programming skills, and the results are, I would say, very convincing also using CAT12. Another advantage is that it's relatively quick compared to FreeSurfer. Obviously, FreeSurfer offers you a lot of opportunities. It's easy to program it. It's very, very flexible. So that's really probably the gold standard. But Cat12 is a great alternative if you're just looking into surface-based analysis and if you want to give this a try. Right. Now let's have a look how Cat12 works. So let's start with the most easiest thing. You want to segment your data and normalize it in order to be able to analyze it statistically. So this is really the first step of preprocessing. Another word of caution, I'm using a really old version of Cat12 from 2019. There are obviously newer versions, but when I was preparing this data at that time, this was done using this older version. That's why I'm showing it using this outdated version. The newer version looks quite similar. It may even be easier to use and smoother to use, but most of the steps are quite similar to that, what I'm going to show you today. So let's click on Segment Data. This opens up a dialog here. Let's wait until it loads. Everything is super slow today on my computer because of the Zoom recording. And it has a lot of settings here. I recommend you to really leave most of the settings unchanged because most of these are standard settings. For example, like a fine regulation, usually looking at European brains. Unless you're in Asia, then you might want to change this. For example, the template is usually quite okay. The voxel size is usually okay. The one thing that you want really to change is to select surface and thickness estimation as yes. You want to make sure that you estimate your surface and thickness data. Otherwise, you won't get surface-based morphometry data. Here you can choose the atlases that you want to extract. For example, you could also choose the Hammers atlas, which is quite popular for epilepsy. And most of the other settings are just all right. And wherever you see an X like this, you need to upload your data. So let's select our data. As I mentioned, we have a number of healthy controls and cases with TLE here. So I'll just go into the folder that contains my data, say select all, and this gives me 126 data points. I load them up into this batch editor, and then I just click this green arrow. It tells me play, and it starts preprocessing of your data. This would take a long time, and I've done this before in order to show you how the next steps will look like. So this is the first step of preprocessing. Right, let's close this. What else do we want to do after preprocessing? There are a couple of other steps you want to do. You want to smooth your data, as I mentioned to you. So for smoothing of voxel-based data, usually I just use the typical smooth function from SVN. And for that, you choose the data that has been generated. That has been generated by Cat12. It's in a folder called MRI, and those are these MVP1 data sets. So modulated, normalized, I think, registered data sets. And P1 is in that regard. It's the gray matter data set, the gray matter tissue data set. Whereas if you look at P2 down here, that will, as far as I remember, be white matter data set. So MVP1, that in this regard you can filter it out, and here we select all this MVP1 data up until here. Right, so we have 126 data sets. These are the pre-processed data sets. For voxel-based morphometry, usually you use something like an 8mm full width at half maximum kernel for smoothing, and you just run this again, and that's it for smoothing. What if you want to smooth surfaces? First, you need to resample them to get the thickness data in these data sets, and then you want to smooth them. And here you again just run this button from the computational anatomy toolbox to resample and smooth your surfaces. So before I was working with voxels, now I'm working with surfaces. And here you load up your left surface data, and really you can just run it as it is. You can here choose the size of the mesh that you want to use. The free surfer mesh is a 164K mesh, so it's a larger mesh. It has many more points on the mesh, which is great if you're looking into effects that are really, really small. But usually it's okay to use the Human Connectome project mesh, which is a much smaller mesh, because it has a much smaller number of comparisons. And if the effects that you're looking for are not tiny, you will also probably see them on this mesh size. And that may improve your statistics because you have fewer comparisons, and then you have a smaller problem with multiple comparisons. Now, looking at what sort of smoothing you want to use, some people like 15 millimeters, some people prefer 12 millimeters, depending on who you believe. And then again, once you've loaded up your data, you just run it, and this will take some time to complete the operation. What else do you want to do for data pre-processing and quality control? You want to control the quality of your data sets. And in order to do that, you can use these buttons here under Check Data Quality. And this is described in detail in the Cat12 manual. And I really encourage you to do this in order to find outliers in your data sets. Outliers may be scans or images where the pre-processing didn't work as properly as in others, or these may be scans where it actually failed. And it's important to find these outliers because if there are mistakes or large mistakes in pre-processing, these may massively influence your results, your statistical results. So it's important to check these outliers and check whether they are realistic or whether there were any errors or issues with pre-processing, and if necessary, exclude these people from statistical analysis. So in our group, we didn't have any relevant outliers, all went well. And now let's have a look at statistical analysis. For statistics, for voxel-based morphometry, you use typically these buttons here on SPM, where you define a statistical model, then you estimate a statistical model, and then you display the results. So when we click at this button, it shows you, again, a batch editor here. Right, and I've already created a model for our group, which is here in results in the VBM group-based results. Let me load it into here. And because I'm comparing two groups, I've chosen for demonstration purposes the most simple model, which is a two-sample t-test. So you have one sample, and those are the 78 files or 78 scans of people with mesial temporal epilepsy on the left with hippocampal sclerosis, and group 2 are 48 age and sex matched healthy volunteers. So these are the two groups. They are independent, and you may add a covariate, which does make sense or is definitely recommended for voxel-based morphometry. And you may include age and sex as covariates, for example. But what you really need to include is total intracranial volume. Why is that? Because people with a larger head size will also have more gray matter in their brains, or a bigger volume of gray matter in their brains. So you need to adjust for this. And this can be done using covariates. Then you can do some masking here, like here, and that's about it for the model. So it's a very, very simple model where we really just compare two groups and have only one covariate. Next, you can go to model estimation. And here you use the SPM math file generated by the first step of the statistical model. And you just estimate the model using a classical method. Next, you can create contrasts. Again, you take the SPM math file that has been generated in the previous step in model estimation. And I've created two contrasts here. One is looking into areas that are bigger in left temporal epilepsy compared to controls. And because temporal epilepsy is the first group, so this is 1 and -1 is the healthy controls. So this is how you code the contrast. And if you look into the opposite, which is actually, which makes more sense, so that there is atrophy in temporal lobe epilepsy compared to controls. So this is left temporal lobe epilepsy smaller than controls. Then you code it as -1 and space 1. So that's how you code this contrast. And this will be the main contrast that we'll be looking at. So atrophy in temporal lobe epilepsy compared to controls. And finally, you again take the SPM mat file that you've created in the previous step, and you just generate the results. And here, if you want, you can choose the statistical threshold and so on. When you just run this, you will get the first nice results. So let's have a look how a voxel-based morphometry result looks like. We go to results, the VBM group results, we load this SPM mat file. And then SPM will ask you which contrast you want to look at. You want to look at atrophy in left temporal lobe epilepsy, so it's this one here. And again, here you see the coding -1 and 1. It asks you whether you want to apply any masking. No, actually, no masking is needed additionally. It asks you whether you want to adjust p-values for multiple testing. So as a first step, I won't do this, but I will show you how to do this in a second step. So that's no. And let's do an uncorrected analysis using a p-value of 0.01. Or 0.001. And then it also asks you what is the maximum threshold of voxel that you want to include. There are a lot of discussions which threshold to use. Maybe say at least 20 voxels need to be in a cluster in order to be reported. So anything that is below 20 voxels probably are very, very small clusters, and that may be noise probably. And this generates an image that is called the glass brain. It looks like this. And you have a lot of significant clusters because we didn't correct for multiple testing. And you have this one cluster on top here, which is highly significant. Right. Let's try to redo this using correction for multiple testing. Again, let's load this SPM mat file. We want to look at atrophy. No masking. Yes, now we want to adjust for multiple testing. And the most conservative approach is using family-wise error correction. So let's click that. And this will be a corrected p-value below 0.05. So this is standard for reporting statistical results in neuroimaging. Again, let's choose 20 voxels as a minimal cluster size. And boom, now you have this well, actually, those are two clusters, one smaller cluster and one bigger cluster. And it reports this on a glass brain. You maybe want to label it using an atlas. Now we're using the neuromorphometrics atlas. If we click on this, where is the maximum? It actually tells you left cerebral white matter, that it's very, very close to the hippocampus and to the thalamus, right? And if you look at this maximum here, that's the left hippocampus, and the other maximum here, that's the left thalamus. What about this? That's well, that's also somewhere over there. But as you can see, it's really hard to see where these areas are using this data set. Now we can also again use computational anatomy toolbox 12 to display our results. We could use the slice overlay feature down here. Let us see what it does. So now first we need to go into the folder where we have our results. It also makes sense before you do this to generate to export the statistical findings from this model, which you can do using the save button here. And I'll have this here. So this is the left TLE is smaller than controls, 0.05 p-value, and family-wise error corrected value. So we just choose that and the statistical result that is being shown here. We want a coronal orientation of our results in this regard. The column chat is just fine. This is all right. Now let us select the slices with local maxima. Okay, so this would, I'll show you two slices. Let's add a couple more slices. So let's say -40, -20, oops, -10. Maybe change this to -5, 0, and 5, and maybe add another one here, which will be -50. So let's do it like this. It asks you about the number of rows and columns. Any that you choose can be used here. And this shows you the results as slices. And this is relatively nice to report your results. So you can see this big, highly significant cluster here in the hippocampus on the left side. It makes sense. So these are subjects with left temporal epilepsy with hippocampal sclerosis. So VBM nicely detects hippocampal volume loss in these subjects, and also mainly affecting both thalami a bit more on the left and on the right. So this is a nice result, but it didn't show anything in the cortex. And my personal experience that voxel-based morphometry is quite good for results in the subcortical structures, but surface-based morphometry is much more sensitive to results based on the cortex. Right, and these are the slices that we've generated. So let's look at surface-based morphometry. In order to run surface models, you use this button here, estimate surface models. Oops, sorry, no, actually you need to first do a basic model, of course, where you define your statistical model. And this actually works very, very similarly to the voxel-based morphometry analysis that we've shown you before, with the difference that typically, you do not need to use total intracranial volume as a covariate for surface-based analysis. There is some discussion about that. I usually do not use this for my research. Right, so then you sort of define for surface-based analysis your basic model, then you can estimate it, and then we can display results. Let's see whether this works. It has been some time, 4 years since I've generated the results, so a lot has changed since then. Let's try to display the surface-based results here. So I will go to my surface-based morphometry group results, and this is the result left clearly smaller than controls, corrected to p below 0.001, so uncorrected results. And we wait a couple of seconds until it loads. And I can already tell you that this only detected a few small clusters. So even though we had a large number of subjects, 126 subjects, I believe, altogether, still the statistics wasn't quite robust enough to show you these very nice large clusters. And you only see these very, very small clusters coming up with analysis based on the full surface. And again, one of the reasons is multiple comparisons. So you only get these very tiny small clusters. They are in those areas, as I've shown you at the beginning. So they affect the temporal lobe. This is, I believe, the posterior part of the insula and some parts of the frontal lobe, but they are very, very small. So what else could you do? As I've mentioned, you could look at regions of interest-based findings. And I've done this here. So we have regions of interest-based findings, and we can load the findings from the left and from the right hemisphere. So these are uncorrected findings using regions of interest. So a word of caution again, these are uncorrected findings, so you will have some false positives. But it's interesting to at least explore these results because they will show you sort of what you see under the surface of your statistical threshold. So let's wait for a couple of seconds until the data loads up. Now this is really, really slow because Zoom is taking up all the energy of my computer. But it hopefully should show up in a couple of seconds. There you go. And sort of these are the uncorrected p-values for your regions of interest. And this already looks more like the data that I've shown you at the beginning from the Enigma study. So again, it affects both frontal lobes and both temporal lobes, with a small predominance for the left side, so for the ipsilateral side. And in red, orange, and yellow, you see the most significant areas, and blue are the less significant areas. So this is the insular cortex and the, I think, precentral cortex. Right. Now let's have a look at the threshold data. using false discovery rate. That's a bit less conservative than family-wise error, but it is also another method to correct for multiple comparisons. Let's wait for the data to load. And this will only show you the most significant results. And those results will have fewer or no false positives. And this is something that you would be able then to report in research paper. Probably I wouldn't properly report uncorrected results because there's the risk of false positives, but you may show those results in supplement. And now here you see nicely these results here displayed on the surface. Maybe you want a white background, which is very easy, and boom, now you have a figure that you could straight away use for your paper and for displaying the results in your paper. Maybe you want to use an inflated surface because some of the results are here sort of deep inside or beneath some of the gyri, and it's difficult to see the insula here. So let's see what happens if we use an inflated surface. It looks like this. Right. Okay. I hope that I could show you some basics of voxel-based morphometry and surface-based morphometry. I hope that this was a fun presentation and that you can replicate some of these results using your own data. If there are any questions, please let me know. Happy to discuss them during the Q&A session. And I wish you a lot of success with these methods during your research. Thank you and bye-bye.